Dear students, last time we discussed about the structure of enzymes and what are the apoenzymes, holoenzymes, cofactors, and what are the coenzymes and prosthetic group activators and so on. In this lecture, I will not take so much time for you people. Look, what is mechanism of enzyme? We are going to discuss about it. How the enzymes act. Okay, enzymes, I told you people, either they break down certain substrate or they combine two substrates together. Okay, so here is one example. I told you people in the last lecture that the enzymes are what the globular protein molecules. They are globular. Okay, and the they have certain part which is known as what the active site. What? Active site. The active site, for example, this is say the active site of this enzyme. Active site. Okay, this one. This whole is what? The active site. But the active site is maintained by all the other amino acids present in the globular structure of this molecule. This is, for example, the substrate. I have drawn here the supposed structure of a substrate. Here, the enzyme and substrate, they combine together. Okay, and this combination is what that is carried out by the part of active site which is known as what the binding site. What you can say, this is what the binding site, this one. Okay, this one is the binding site. The binding site has the capability to bind the substrate, to combine, to form the bonds with the substrate. And then after its combination with the substrate, the catalytic site, this one is say the catalytic site, this one. Okay, now you can say here was the binding site and the catalytic site together. Okay, active site means what? The binding site plus catalytic site. It is equal to what? Binding site plus catalytic site. Okay, so these two parts are important. As you see that there is a small part of your pen that works. See, here is your pen. Okay, and its a small part is active. The other part is what? Its cap, etc., etc., etc. All these are nothing but this part is active. Okay, so this would be active, this would be functional if the other part of the pen is there. If this is not there, what will happen? This pen won't be able to act. This won't be able to function. The same is true with the enzyme. It's active site consists of binding site and catalytic site. The binding site first binds with the substrate, this part and this part, and then the catalytic site carry out the catalytic activity, either binding to say this is one substrate, this is another substrate, and they bind together. Okay, as they bind, that is what the catalytic activity. Here the catalytic activity of the enzyme is breaking. Okay, it has cleaved this substrate into two parts. Okay, let us suppose this were what the maltose. This would be glucose molecule, this would be glucose molecule. The enzyme is then separated. Okay, these are the products, and this is what the enzyme. This is scenario A. Scenario A, where the enzyme acts upon substrate and products are formed, and that's how story ends. There is another story of enzymes action. That is what? This is, suppose this is A. Here is a B story. Okay, and this is suppose story B. This is what? There is an initial substrate that is acted upon by an enzyme. Enzyme, say this is enzyme 1. You can call it enzyme 1. The product would be formed. This would be intermediate product. Then on this intermediate product, the enzyme 2 would act. Then this product is formed. This product is acted upon by another enzyme. That could be what you can name it, enzyme 3. Enzyme 3 acts upon C and forms what? T. Okay, it's again an intermediate product. Okay, it is acted upon by what? Enzyme 4. Okay, and what happens? The final product is formed. As the final product is formed, you can say the final product means that the objective has been achieved by the cell. Okay, now this final product will send the message to enzyme 1 that no more catalysis is required. Okay, one thing. Second is what? That initial product, as it comes, initial product, it stimulates the system. What? It stimulates the systems to carry out to speed up the reaction. For example, here is what, say this is the initial product, initial substrate. Sorry, it would act upon here. That product should be formed. Final product should be formed. So this is the promotion. As the initial substrate is available to the cell, what happens? It promotes the final product formation, and as the final product is formed, it inhibits the beginning or start of this cascade system of the biochemical reactions in the series. So this is what the enzyme mechanism of enzyme actions. Okay, there are two models. There are two models of enzyme action. One was put forward by Emil Fischer, and the second was put forward by Koshland. Okay, we'll discuss the Emil Fischer model and Koshland model in the next lecture.